The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hi children, how are you? Hope you are healthy and happy. Today I am going to begin the session by saying a quote. Do it now, sometimes later becomes never. Dear children, you may be knowing the word procrastination. Yes, it is the biggest slayer of the mind. Do not postpone anything which hold importance for you. A task, for example, examination. It seems difficult for you in the beginning, but if you plan properly and if you are taking effort for it, surely you will be able to complete it successfully. What you have to do is just take the first step, and once when you put the wheels in motion, surely you will be able to accomplish the task and you will be happy in your life. Okay, dear children, so this is what I want to say. Please don't postpone anything which is important for you. By saying this, I move on to today's session. In the today's session, we are going to first recap the previous class and then we will be moving on to the next topic. So, in the previous class, we started with the new lesson, Unravelling Genetic Mysteries, and in this chapter, we understood that the children acquire certain features from their parent and certain features they are showing which will be different from their parents, and it is due to the term which is called genetics, isn't it? So, in the previous class we studied that the transmission of features of parents to offspring is termed as heredity, and the features seen in offsprings that are different from their parents are called variations. And the branch of science that deals with heredity and variation is called genetics. And then later on we understood the emergence of genetics. Gregor John Mendel was the scientist who conducted hybridization experiments on the pea plant and led to the foundation of genetics. Therefore, Mendel is considered as father of genetics. In his experiments, he done it on the basis of two contrasting traits of the character height in the pea plant. So, first experiment which he conducted in the pea plant was based on the contrasting traits of the character height, and in that he took the factors by using the symbol, and this one we have seen. And we came to know the experiment. You can see here parental plant, that is tall plant and dwarf plant. The tall plant, he has given the symbol as capital T, and dwarf plant he has given the symbol as small t, isn't it? And these we already studied that these symbols, they are the alleles. A gene has different forms that are alleles, isn't it? And it has been said that a gene has two alleles, for example, capital T and small t are the different alleles of gene that controls the character height. So, here we understood these are the factors or the alleles of a gene, and he took it in the form of a symbol as capital T for tall plant and small t for the dwarf plant, isn't it? Now, during gamete formation, from the pair of this symbol for tallness, only one capital T has involved in the formation of gametes, and from small t only one small t has involved in the formation of gametes, and later these were fused, and after the fertilization Mendel got a plant which is tall, but in this tall plant the factor capital T and small t was present. So, this is the first generation plant which was seen or which was found tall, but in this tall plant the factor capital T and small t is present, isn't it? And the indicator we discussed based on the first experiment, and later we understood that when a plant that differ in a pair of contrasting traits are hybridized, only one trait is expressed while the other remain hidden in the offspring of the first generation. The expressed trait is called dominant trait and the hidden trait is called recessive trait. So, from the first experiment we understood what the tall plant was appeared in the first generation, isn't it? In the first generation tall plant was appeared, and as small t is hidden, it is not expressed there. So, this small t is the recessive trait, okay, and tall plant it is expressed, that is capital T is expressed here, that is why the plant become tall, that is why we can say it is dominant trait. So, from this he concluded by giving law of dominance, and the law of dominance states that the trait which is expressed is dominant trait and the trait which is hidden is the recessive trait. Then later on he was in dilemma, what happened to the small t, isn't it? The fact that no intermediate plants were formed by the hybridization of tall and dwarf plants, and the factor responsible for dwarfness remaining hidden persuaded Mendel to continue with his experiment. So, later on Mendel again continue with his experiment. So, what he done in the next step? He self pollinated the first generation plant, and he self pollinated, it is been said. He took the first generation plant which have the symbol capital T small t. This is the first generation plant which is having the symbol capital T small t. He took that plant and he self pollinated that plant, okay, and you can see here this is the result which he got after the self pollination of the first generation plant. So, he took capital T small t as a parental plant, crossed with another capital T small t, and the gamete of this parental plant are capital T small t for this parental plant, and for this parental plant it is again capital T small t, and later on after the fusion, after the fertilization, he got the plants or he observed the plants there as tall plant, tall plant, tall plant and one dwarf plant. But in this tall plant you can see pure capital T factor is present there. In this tall plant one capital T and one small t factor is present there. In this tall plant one capital T and another small t is present there, and in this dwarf plant the pure small t is present there, and based on this he took the ratio, and when he took the ratio he found that there are 3 tall plants as capital T is expressed here and small t is hidden in these plants. So, dominancy is with the capital T, therefore, the plant is tall, and he got the ratio as 1, 2, 3, 3 tall plant and 1 dwarf plant. So, 3:1 ratio he got from the self pollination of the F1 generation plant. So, that was the experiment he conducted based on the character height. I think you would have understood this part very clearly, my dear children. Indicator I told you to do it, and next thing again he conducted more and more experiments on the pea plant by taking the position of the flower, shape of the seed, and in such kind of parental plants he got the same ratio as 3:1, 3:1. And next he was having a question, why does the recessive trait in the first generation appear in the second generation? In the first generation that recessive trait was hidden, but in the second generation it appeared, isn't it? The small t, small plant, dwarf plant we have seen in the second generation. Why it happened? Later on he came up with the assumption that during gamete formation, the factors that determine a particular character segregate, that means they separate without getting mixed. That is, half of the gametes formed from first generation plant capital T small t contain capital T and the other half contain small t. So, from this again he concluded and given the second law of inheritance, and that was law of segregation. In the law of segregation he stated that during gametogenesis, gametogenesis means during gamete formation, the pair of factors they will segregate from each other, that is they will separate from each other, and it has been said that half of the gametes formed from the first generation plant contains capital T and the other half contains small t. That is how the mixing of the gamete is taking place. So, that separation is the law of segregation. I think you would have understood the second law of inheritance given by Mendel, and based on this experiment he came with the conclusion, isn't it? Based on the first experiment, the inference formulated by Mendel, inference means conclusion, formulated by Mendel was a character is controlled by the combination of two factors. One trait is expressed which is dominant and the other trait remains hidden, that is recessive in the first generation. Number 3, the trait that remain hidden in the first generation appears in the second generation. In the first generation the trait which was hidden, it was dwarf plant, isn't it? The dwarf character was hidden in the first generation, but it appeared in the second generation, and the ratio of the dominant and the recessive trait in the second generation was found as 3:1. So, that was the conclusion given by Mendel from the first experiment based on the character height. Now, later on it has been said that in his first experiment Mendel observed the inheritance of a single pair of contrasting trait. So, first experiment he conducted on the pea plant was based on only one character, and that was the height of the plant. Only he focused on the height of the plant and he conducted the experiment. Now, he observed the inheritance of two pairs of contrasting traits of the same plant in the next stage. Next, what he done? He done the second experiment, and in the second experiment he took two contrasting trait. Okay, you can observe the illustration which is showing the hybridization experiment conducted by Mendel on two characters, namely height of the plant and shape of the seed. So, now in the second experiment Mendel has taken two contrasting characters. One is the height of the plant, number 2 is the shape of the seed. So, by taking these two characters he conducted the experiment on the pea plant. So, we can see the illustration, okay my dear children. So, you can see here what is happening. This is the parental plant where he took the height of the plant. One is tall plant, the other one is dwarf plant, and number 2 he has taken the second character there, that is the shape of the seed. It is round seed, and here again he took the second character here as wrinkle seed. And the symbol for the tall plant is capital T, and the symbol for the round seed is capital R, capital R. It is crossed with dwarf plant which is small t, and wrinkle seed he has given the symbol as small r, small r. Now, while crossing these two parental plant, gamete formation will be taking place. Already we know that because of the gamete formation, and the law of segregation states that during gametogenesis the pair of factors will segregate from each other, they will separate from each other. So, in this a pair of character is there. In the case of capital T, only one capital T will participate in the gamete formation, and here a pair of factor is present there related to round seed. Among this only one capital R will be participating in the gamete formation. Now, this factor is there for the gamete formation, that is capital T and capital R. Okay. In the case of small t, small r, again you can see only one small t and only one small r is there in the form of a symbol which is a factor which is participating in the gamete formation. Now, these are the factors which will participate in the gamete formation, and later on a fusion will be taking place, and after fertilization we can see there capital T, capital R, small r, capital T, capital R, small r. This is the factors which is present there in the first generation of the experiment number 2, where Mendel took two characters, that is the height of the plant and the shape of the seed. So, the factors capital T, capital R, small r is present there. But what Mendel observed there when the plant was coming up with the height, isn't it? He observed that capital T, capital R is dominant, whereas small t and small r is hidden. Okay. So, the plant which was there, it was tall and it is having a round seed in it. So, we can say here tall plant with round seed, isn't it? Tall plant with round seed plant was observed in the first generation for the experiment, where he took two contrasting characters. So, this is important, my dear children. In this case, tall plant with round seed is dominant, dwarf plant with wrinkle seed is recessive or it is hidden, it is not appeared there. Okay. So, in the first generation, tall plant with round seed is dominant, it is appeared, and Mendel was able to see that plant as it was tall with round seed. Now, what he would have done? What he has done? He self-pollinated the first generation result. This is the first generation result, isn't it, my dear children? When he self-pollinated the first generation result, you can see here the cross is being done. The experiment is being done based on this result, that is capital T, capital R, small r, when crossed with capital T, capital R, small r. Surely, first we have to take the gametes from this part, and the gamete of this part is capital T, capital R, that is capital T, capital R, again capital T, small r, again small t, capital R, again small t, small r. So, this is the 4 gametes of this pea plant. From this pea plant, these are the 4 gametes which are formed there. In the same manner, when the gametes were taken from this plant, it is seen as capital T, capital R, capital T, small t, capital R, and small t, small r. So, you can see here the capital T, when crossed with, when it forms a gamete, it will be having a gamete as capital T, capital R, then capital T, small r, then small t capital R, then small t small r. Okay, you can see here small t small r. So these are the gametes of these two pea plants. Now after the formation of gametes, what will happen? Surely fertilization will be taking place, and the result after fertilization which Mendel got is, this is the result. This is the plants. These are the plants which is having these types of factors present in it, isn't it? So you can see here, when capital T capital R cross with capital T capital R, the result is same. You can see capital T capital T capital R, that is tall plant with round seed. Same way, when this capital T capital R is crossed with capital T small r, the result is capital T capital T capital R small r. You can see here, isn't it? The same manner. So here we can see again tall plant with round seed, as this is recessive. Now again this capital T capital R, when it is crossed with capital T small r, so that this result we have seen just now. Again this will be crossed with this one. Surely the result will be capital T small t capital R small r, isn't it? And the same manner here, when this gamete is crossed with this gamete, surely after fertilization the plant will be having this factor in it, that is capital T small t capital R small r, capital T small t capital R small r. You can see here. Okay, my dear children. So that is related to the results of the second generation after self pollination of the first generation plant. So in the same manner, this one will be crossed with this one, again this one will be crossed with this one as this one, and the result you can see here. Same manner, this gamete will be crossing with these 4 gametes, and the result is here. The same manner, this will be crossing with these 4 gametes, and the result is here. Okay. So it is the illustration related to the hybridization of 2 characters. So this is the result where we can see the hybridization of the 2 characters. Now what is the indicator? You can see here, factors present in the gametes produced by first generation. Which are the factors present in the gametes produced by first generation? You can see here, in the first generation what happened? When he conducted the experiment, the factors are of the result, capital T small t capital R small r. This is the first generation result. The factors present in it are capital T small t capital R small r. So that is the answer for the first indicator. Okay. Now characters in the plants of the second generation. Second generation, which are the characters we can see in those plants? So we can see here the characters as tall plant with round seed, tall plant with wrinkle seed, dwarf plant with round seed, and dwarf plant with wrinkle seed. These are the 4 characters we can see in the case of the second generation result, which is being done by the self pollination of the first generation result, isn't it? Again I will repeat it. The 4 characters we can see as a result of the second generation, and they are tall plant with round seed, tall plant with wrinkle seed, dwarf plant with round seed, and dwarf plant with wrinkle seed. So these are the 4 characters we can see in the second generation. Next it is been asked, characters different from parents that appeared in the second generation. Which are the characters which were different from the parents and they appeared in the second generation? So we can see here, see this is the pure parent, isn't it? And this one is also the pure parent, because we know that when we cross the plant, it was capital T capital R small r, small t and small r, isn't it? So surely this one is the pure parent and this one is the pure parent, and the 2 intermediate or the 2 new plant which was observed there in the second generation are tall plant with wrinkle seed and dwarf plant with round seed. So these are the 2 intermediate plants, or we can say these are the two new plants which was observed by Mendel after the self-pollination of the first generation result, and he got it as tall plant with wrinkle seed and dwarf plant with round seed. So, these are the two new plants which he observed after the experiment conducted on the first generation result. I think you would have understood about the characters which are different from parents that appeared in the second generation. One is the tall plant with wrinkle seed, that is capital T small r, and the other one is dwarf plant with round seed, that is small t capital R. So, this is the answer for this indicator. Okay. So, I think you would have understood about that part. Now, through this experiment, Mendel could find the reason for the appearance of new combination of characters in offsprings that were not expressed in parents, isn't it? So, through this experiment, Mendel was able to find out the reason for the appearance of new combination which was not present in the parent. So, with that, Mendel explained that the appearance of variations in offspring, that is the character which is not found in the previous generation, isn't it, is due to the independent assortment of each character, This is the reason for the explanation of the appearance of variation in the offspring. Understood, my dear children? So, Mendel said that because of the independent assortment taking place in the character, because of that only variations are taking place, and we know that what are variations? Variations are the characters which are not seen in parents, but they are seen in offspring. So, the variation is taking place because of the independent assortment of each character. So, from this again Mendel concluded and gave the third law of inheritance, and that was law of independent assortment. That means the characters which were present there, isn't it, in the first generation during the crossing, isn't it? Here you can see capital T, small capital R, and it is crossed with small t, small r. This was the characters, but later on what happened, we can see here capital T, small t, capital R, small r, and after the self pollination what we have seen there, capital T, small r is another character, small t, capital R is another character. So, these two characters, they are the new combination or new characters which is formed there, isn't it, and it happened because of the independent assortment of the character. That means tall plant is not depending on round seed, as well as dwarf plant is not depending on wrinkle seed. They are independently separating from each other, and they are combining together or fusing together for the formation of new characters. So, that is why it is called independent assortment of characters. I think you would have understood about independent assortment of characters. Okay, my dear children, again I will tell you, in the second experiment when Mendel took tall plant and round seed, and when he crossed this plant with dwarf plant with wrinkle seed, he was able to see the varieties of plants other than parent plant, and it happened because the factors which are there for tall plant and round seed, dwarf plant and wrinkle seed, they were not together, they were separated from each other, and they fused with the other character and they formed a new character there. That is why a new combination was seen there by Mendel after the self pollination of the first generation, when he came to know in the result of the second generation. He understood that because of the independent assortment of each character, these new characters were formed. Understood, my dear children? That is what Mendel explained for the appearance of variation. And later on Mendel said that Those which are responsible for the inheritance of characters are factors, isn't it? The characters which were formed there, the characters which are seen there in the offspring is due to the factors present in the character, and the real structure and peculiarities of these factors were not identified till early 20th century. And later on, when the studies and the significance of DNA came into existence, that time the inheritance of character was made clear. Later on, the studies on DNA, that is deoxyribose nucleic acid, and its significance was clear there. That time it was understood the relation of inheritance of character, isn't it? It was also found that the carriers of heredity which Mendel described as factors were the genes present in the DNA. Okay. So, based on this thing, the scientists, they done the research on the structure of DNA, that is deoxyribonucleic acid, and the scientists were James Watson and Francis Crick, and they presented the double helical model of DNA in 1953. So, when the research on DNA and its significance was being conducted, that time only it was clear that inheritance of character is connected with DNA, and because of that the two scientists James Watson and Francis Crick, they presented a double helical model of DNA in 1953, and they got Nobel Prize in 1962. And in the structure of DNA, they said that DNA molecule contain two strands. It is having, this is a structure of DNA and it contain two strands. This is 1 and this one is 2. The blue color is first one strand and the red color is the another strand. So, the DNA contains two strands, a structure with two long strands made up of sugar and phosphate and rungs with nitrogen base. It is a structure where two long strands are there and it is made up of sugar and phosphate, and these are the rungs which are having the nitrogen base. You can see here this blue color adenine, the red color is thymine. These are the two base pairs, isn't it? Adenine always pairs with thymine, and yellow cytosine, the green color is the guanine. Always cytosine will pair with guanine. You can see the structure of these nitrogen base pair. These structures you can see with the structure, only the adenine will be able to pair with thymine, only the cytosine will be able to pair with guanine. So, this is the long strand which is having sugar and phosphate in it, and the rungs, you can see these are the rungs, and these rungs, they are the nitrogen bases which constitute the adenine and thymine, guanine and cytosine. So, you can see here this is sugar, this is the phosphate, this is the sugar, and here this one is the nitrogen base. Okay. So, this is how the structure of DNA is being. See, you can see the one of the structure, isn't it? Here this part only, you can see this part little bit more elaborated. Here you can see it has been said phosphate, sugar, nitrogen base. Okay, it is an illustration of a nucleotide. You can see here DNA molecule is made up of units called nucleotides. So, it is made up of molecule called nucleotides. You can see here these long strands, it is made up of sugar and phosphate. This is phosphate and sugar, phosphate and sugar, phosphate and sugar, they are connected with each other, isn't it? And this is the one of the unit which is called nucleotide, and like this many units of nucleotides are seen in the structure of DNA. Many nucleotide, that is phosphate, sugar and nitrogen base constitute a nucleotide in the structure of DNA, and a complete structure of DNA constitute many nucleotide which includes phosphate, sugar and nitrogen base. So, I think you would have understood, my dear children. So, what is nucleotide? Nucleotide is the units of DNA present in the DNA structure, and a nucleotide contains a sugar molecule, a phosphate molecule and a nitrogen base. This is very important. You have to study it. It used to come in the question paper, my dear children. This structure is very important. You have to study it. Okay. So, this is related to the nucleotide, which is a unit present in the DNA molecule, and later on you can see here DNA contain a sugar we have said, and the name of the sugar is deoxyribose sugar. DNA, in the DNA structure a sugar is present there, and the name of the sugar is deoxyribose sugar. And the nitrogen bases are the molecules that contain nitrogen and are alkaline in nature. We already said that the rungs in the structure of DNA is nitrogen bases, and what are nitrogen bases? They are the molecules which contain nitrogen and they are alkaline in nature. Since DNA has 4 kinds of nitrogen bases, already we understood that DNA is having 4 kinds of nitrogen bases. Number 1, adenine, thymine, guanine and cytosine, isn't it? That is adenine, thymine, guanine and cytosine. Therefore, DNA has 4 kinds of nucleotide too. So, in the structure of DNA, 4 kinds of nucleotides will be present there. One is adenine nucleotide, number 2 is thymine nucleotide, number 3 is cytosine nucleotide and number 4 is guanine nucleotide. So, in the structure of DNA, how many types of nucleotides are there, my dear children? 4 types. Okay, my dear children, adenine nucleotide, thymine nucleotide, cytosine nucleotide and guanine nucleotide. This name is been there because of the presence of 4 nitrogen bases, and they are adenine, thymine, cytosine and guanine. Next you can see nitrogen bases. The building components of DNA are molecules with great specificity. In DNA, the base adenine pairs only with thymine and the guanine pairs only with cytosine. This part is again important. It has been said that in the structure of DNA molecule, nitrogen bases are the building components because adenine will be always pairing with thymine because of its structure, and the same way cytosine will be always pairing with guanine because of its structure. So, this is the specificity we can see in the structure of DNA, which is again very important. I think you would have understood about the structure of DNA. So, indicator, components of nucleotide. What are the components of nucleotide which we have studied just now, my dear children? It is phosphate, sugar molecule and nitrogen base. This is the components of nucleotide. And what are the nitrogen bases we have studied? The nitrogen bases are adenine, thymine, guanine and cytosine. And the specificity maintained by nitrogen bases in pairing. What is the specificity which is maintained by nitrogen bases in pairing? Adenine pairs with thymine and guanine pairs with cytosine. That is the answer for this indicator. I think you would have understood about it clearly. Next one you can see, the next illustration is related with the strands of DNA. This is the illustration which is related with the strands of DNA, and this illustration is very important as it used to come in the question paper for completing the strand. That is one part, the one side is given there, the other side you have to draw. It has been said there, the arrangement of nucleotide in a DNA molecule is illustrated below. Redraw this illustration, isn't it, and complete its second strand. So, this one you have to draw in your file, my dear children. The first part or first nucleotide related to the DNA structure is given there. As an example, you can see phosphate, sugar and guanine. Guanine we know that it pairs with cytosine. Cytosine again this is sugar and this is phosphate. The same manner you can see here. Now again you can draw, again here one line you have to draw, and this connection you have to give with the next one, that is the sugar molecule. And the second part, the sugar molecule is connected, it is being joined with the thymine, and we know that thymine is having the pair with adenine. So you have to draw the structure of adenine, nitrogen base, and with that you have to draw these two, that is the sugar and the phosphate molecule. Again in the third you can see cytosine is given there. Surely cytosine is the nitrogen base which is pairing with the guanine. So you have to draw here the structure of guanine. With that you have to draw the structure of phosphate and sugar molecule. So this, like what is being shown here, the same you have to shown here with the, by drawing the structure of phosphate and sugar molecule. Okay, and the last one is adenine. It is pairing with the thymine. So you have to draw the structure of thymine here, and the thymine is connected with the sugar molecule and the phosphate molecule. In this manner the second strand will be completed. You have to draw this one in your file, my dear children, and you have to send it to me through WhatsApp. Okay. So that is related to the strand of DNA. Now as it has been said that DNA is the molecule which is having gene in it, and gene is a factor which is controlling the characters which is been transmitted from one generation to another generation. So DNA is deoxyribonucleic acid. It is a type of nucleic acid. In the same manner there is one more nucleic acid, and that is RNA, which is also known as ribonucleic acid, and RNA is another nucleic acid like DNA. RNA is also formed of nucleotides. Ribose sugar is present in RNA. In the case of DNA, name of the sugar is deoxyribose sugar. In the case of RNA, the sugar is ribose sugar. And in RNA, nitrogen base uracil is seen instead of thymine. In the case of nitrogen base present in DNA, we have studied that adenine and thymine, they are the nitrogen base, and guanine and cytosine is the another nitrogen base present in the DNA structure. But in the case of RNA, the nitrogen base uracil is present instead of thymine. So in the structure of RNA, the adenine will be pairing with uracil instead of thymine. In the structure of RNA, adenine will be pairing with uracil instead of thymine. Okay. And next one, it has been said that majority of RNAs have only one single strand. We have seen that DNA is having two strands. It is double helical structure, isn't it? It is having double helical structure, and in the case of RNA only single strand is present there. Okay. In the case of RNA only single strand is present there. We can compare the structure of DNA and RNA, and you have to complete this table also. While comparing the structure of DNA and RNA, you can see here number of strand, type of sugar and nitrogen base. DNA, in the case of DNA, the number of strands are 2. In the case of RNA, the number of strands is 1. In the case of DNA, the type of sugar is deoxyribose sugar. In the case of RNA, it is ribose sugar. In the case of DNA, the nitrogen bases are adenine, thymine, guanine, cytosine. In the case of RNA, the nitrogen bases are adenine, uracil, guanine, cytosine. I think you would have understood the structure of RNA also, my dear children. With this, I wind the session here. If you are having any doubt related to this session, my dear children, please call me and clarify your doubt. Please don't postpone the work for tomorrow. I told you why, because sometimes later never comes, isn't it? Sometimes when we postpone the work for tomorrow, it will never come. Again and again it will be, we will be postponing it for the next day. So In that manner, what will happen? The work will be pending there, the doubts will be pending there, and we will be having lot and lot of problem, and we will not be able to move to the next step. So, my dear children, when you are studying, when you are understanding, if you are having any doubt related to today's session, please give me a call. So, my dear children, I wind up for today's session, and we will be meeting in the next session. Till then, take care.